All right, before I even get started, I'd just like to say thank you so much for giving me this opportunity. I've seen like how big your guys' chapter has gotten within this last year. And it's honestly such an honor to be able to present to such a large group. So yeah, this is just going to be a short presentation on my experiences within miRcore in the last few years. So here's just a brief overview of everything, and I'll go into more depth of everything in the next few slides. But essentially, I first got involved with miRcore 2 years ago in 2022, and essentially I started by taking the computational biology camp, which then led me to take the miRcore volunteer program throughout the year, and finally I started a GIDAS branch within my own school. So to start off with the miRcore summer camps, there are three of the camps that I've taken. And the first one I started with was the computational biology camp through miRcore. And it was essentially a camp that focused on the use of computational tools such as GO2R, SpringDB, KEGG pathways, and gene cards. And I'm sure you guys are familiar with them like through your own GIDAS club. But essentially that camp kind of explored using these tools to focus on like a mini research project. So in my instance, I did a little research project on melanoma or skin cancer using all of these computational tools. So it was a really good way of kind of getting introduced to all of these tools, and it kind of brought me down the path of getting more involved with miRcore and pursuing more research within this organization. The second camp I took was the R and machine learning camp, and this camp kind of explored the use of the R programming language, which is a statistical language, to kind of visualize different genetic data. So we learned how to visualize heat maps, box plots, and other types of graphic visualizations through the use of this programming language. And it is with genetic data, so it kind of tied back with some of the research we were doing previously. And then lastly, we used a bit of random forest to kind of explore the field of machine learning through R itself. And then the last camp I took was the biotechnology sequencing camp, and that kind of taught us how to use the Linux operating system in processing and understanding next generation sequencing data. So, for example, we used Bowtie2, SAM tools to kind of process NGS data and make them more understandable. So these are kind of all the summer camps that I went through. And they've definitely helped in creating a good foundation in like any types of research and kind of the standardized miRcore research process. So I'll get more in depth on that later too. So after taking the computational biology camp, I got involved with the miRcore volunteer program in the same year, 2022. And as um, as for any student trying to get involved with the miRcore volunteer program, you do need to take a miRcore summer camp. But essentially, the MVP program focuses on conducting research throughout the year. So individuals go through research like every single week from the beginning of September to all the way to like April. And each year we have an annual disease of focus. So there's a subset of diseases that we can research within that one year. So for example, within 2022, we did research autoimmune diseases or diseases involving the immune system. And then within this last year, we did neurodegenerative diseases. And I'm sure you guys are familiar within your own GIDAS clip. You might have done some research in that area. And for the next year of miRcore, it's most likely going to be neurodegenerative diseases as well, just to kind of get more involved because we've only touched the surface level within this past year. So as of now, that's what we're thinking, but it does need to be decided. And then lastly, through the miRcore volunteer program, we did an extensive amount of preparation for the Genes and Health contest, which is kind of an exam that individuals can take in MVP that kind of assesses your ability of using computational tools, different programming languages, and essentially all that miRcore has taught you within the last year. So these are some of the things that I was involved in, like specifically through the MVP program within the last year. So just to go a little bit more in depth on some of the past research projects that I worked on within MVP, in 2022 to 23, it was the autoimmune disease subset for the annual disease of focus. So in my group specifically, we worked on the role of cholesterol metabolism within Crohn's disease, which is an autoimmune disease. So throughout the year, like I said, you will be working, it is in a group setting. So you work with a group to kind of focus on one disease that's within that subset of diseases. So for the last year, I did work on Crohn's disease specifically, and I looked at cholesterol metabolism within the digestive system and how that plays a role in the progression of the disease. And then in this past year, I looked at frontotemporal dementia, again, within the neurodegenerative disease subset, and kind of looked at how oxidative phosphorylation played a role in that. And if you guys are curious, I can go more in depth on that during the question session. And then more recently, I started getting involved with the miRcore RNA sequencing research, which is separate from MVP. It's conducted on different days, but essentially every week we met and kind of we actually, and the thing that differentiates between MVP research and RNA sequencing research is in RNA sequencing research, we do novel research, stuff that hasn't been done before. So essentially, we were trying to create new discoveries within scientific field, and specifically, we focused on understanding the commonalities between different transfer RNAs in Zika virus, as well as their functions. And once again, I will go more in depth on that if that's something you guys are interested in hearing about. And then, as I mentioned before, I did join, after I got involved with miRcore through the summer camp, I did eventually go on to create a GIDAS branch within my school. So in 2022, I did create GIDAS branch, and just like how you guys are doing, I've seen how well you guys are doing it though. I mean, you guys were the top school for fundraising and everything, so congrats on that. A lot of so proud of you guys. It's a lot of work to do that, so a lot of respect for all of the commitment and dedication you've had within the past year. But essentially, I did what just generally any GIDAS branch does. I taught like the use of computational tools and research to all of the members. And then we kind of prepared for the Genes and Health contest, which I mentioned before, was just an exam to assess your ability of using these computational tools for research. And then lastly, in our club, we did focus on doing the research itself and presenting for the annual high school GIDAS research conference, which was held like last month. So it's pretty recent, all of this. And I want to just say, I know I've gone through all of this and talked about how much I've been involved with miRcore. And I understand there's a huge time difference between the US and Korea. So a lot of these opportunities might not be the most accessible for you guys because the time difference is pretty big. So you guys might be staying up in the middle of the night trying to join all of these individual events. So while all of these might not be the most accessible for you, I just want to kind of illuminate on all of the different opportunities present within miRcore, so that in the future, if we are able to change the timings of some of these events, you guys will have the opportunity to participate in it. But I do want to say you guys are already doing so much as a GIDAS branch, even with all of the limitations with the large time difference. So I just want to applaud you on that. And I know that the branch is going to grow even further, and I can't wait to see what you guys do within the next years. So that is pretty much it for my presentation. Thank you so much for listening. If you guys have any questions, please feel free to ask them.